Hello, welcome in this video lecture. So, in this current video lecture, we are going to study the chapter that is enzymes. Up till now, we have studied different biomolecules like carbohydrates, amino acids, proteins, and now this is the fourth biomolecule that is the enzymes. So, whenever enzyme as a definition point of view is considered, no doubt enzymes are simply biological catalysts which enhance the rate of various physiological reactions present in any type of living organism and human being. Hence, these are also called as protein catalysts. Suppose, as the catalysts are the substance that increase the rate of chemical reaction. In laboratory, we are doing different types of chemical reaction. And suppose we have to enhance the rate of that chemical reaction by using chemical compound, then we are going to use specific catalyst. So, catalysts are the substance that increases the rate of chemical reaction. In the same manner, life requires many chemical reactions occur within organisms, right? The human body employs over thousands of reactions regularly, and many of these reactions will occur too slowly to be useful in the absence of catalyst. They occur very slowly. For this purpose, nature provides humans and other biological organisms with a proteins. Particularly, these such kind of proteins are capable of catalyzing the biological reactions, and these specific proteins which is capable to enhance rate of various biological catalysts, such reactions, such compounds are called as enzymes. That is, enzymes are nothing but these are the proteins which are used to increase the rate of reaction. Right? So this is very simple definition. See the slide. Whenever different plants and species are considered, near about 5,000 various reactions are catalyzed by enzymes. These enzymes are capable to increase the rate of chemical reaction by 1,000 of that particular factors are used and 1,000 in this manner. So according to this, particularly the scientist who specifies the studying enzymes called as enzymologist. This is one of the terms. Those scientists which we are going to study the different catalytic reactions of enzyme are called as enzymologist. Right? So this is enzymology is one of the branch which is developed to study such kind of enzymes, and hence enzymologists always refer the reactants of catalyzed reaction as enzyme. Now we are moving. What is the structure or model of enzyme catalyst? How they are catalyzing? Enzymes are containing one important specific part which is having highly reactive or active portion known as active site. Active active site of enzyme means what? See the diagram shown in the slide. Enzymes containing a specific functional group. In the diagram it is shown by two red concaves or curves, and these red curves are called as active sites. Active sites are nothing but these are the particular sites which are having tendency to react with another particular molecule or provide the space to the reaction. So active sites are very important present in the enzymes. Now see, most of the enzymes are composed by hundreds or thousands of amino acids, and whenever they are having hundreds or thousands of amino acids present in the molecule, then automatically okay, we are continuing. So whenever any type of biological reaction takes place, these enzymes present in a smaller region comes in contact with the substrate. Now simple question arises, what are the substrate? Substrates are nothing but these are the biological material which is used for biological process. For example, when we are eating some food material is the substrate used for process of digestion. So the substrate is a biological material which is always catalyzed by various enzymes. So remember the substrate. Then in the enzyme there is an active site. Active site means what? Which is regularly participated in a catalysis of substrate, and these active sites are specific functional group which is actually involved in catalysis process called as active sites. So remember that enzyme contain active site, and substrates are the material which is always comes in contact with various enzymes and their active sites. So see the diagram. Now how the reaction or catalysis takes place we see. Now we will consider the reaction where two substrate, that is reactants, are converted to one product. See the diagram. In the diagram there are shown two substrate. One is shown by orange color, another is shown by parrot green color. Whenever these both substrates are combined, we are getting product. This reaction is shown in absence of enzyme. कशाच्या absence मध्ये दाखवली? Enzyme. This is simple. Now whenever these two substrate comes in contact with enzyme, that is biological catalyst, then the rate of combination of these two substrate react with each other rapidly, and we are getting one product whenever two substrates are combined in presence of enzyme as a catalyst. Reaction takes place like this. See the reaction, how it takes place. Illustration, very important example. Now see the step 1. What is the step 1? Here enzyme with their active site is shown. Our two substrate are also shown. One is the green colored, another one is the orange color. Two substrate. In step first, these both substrate forms complex with enzyme, and This complex is called as ES. Remember the wording, E for enzyme, S for substrate, right? So in first step, there occurs complex formation between enzyme and active site of enzyme and substrate which is to be catalyzed in the reaction. So the role of active site is to provide space to attach substrate on its surface, and this is the role of catalyst, that is enzyme catalyst. So in first step, formation of ES, enzyme substrate complex. In second step, enzyme, on the surface of enzyme both substrate are get attached, and the catalysis reaction is formed in such manner that we are getting combination of enzyme with product. Second complex is formed. First complex is enzyme with substrate, second complex is enzyme with product. So this is the enzyme catalyzed reaction gives formation of enzyme product complex. And now in the third step, we are getting the release of enzyme or release of product from enzyme. How much quantity of enzyme is used for catalyst totally separated from released product, but rate of reaction is get increased, and those enzyme which is separated again used for first step. So the enzyme and substrate reaction takes place in these three steps. See the slide carefully. Step 1, formation of enzyme substrate complex. Step 2, formation of enzyme product complex. Step 3, separation of product from enzyme, and those enzyme which is released again used for step first for new reaction. So in the step 1, substrate bind to active site of an enzyme. The substrate are held tightly in active site by non-conventional attractive force which are maximum due to complementary slabs of substrate and active site. They gives formation of enzyme substrate complex. In second step, the chemical reaction occurs on the surface of enzyme, substrate is converted into product, and coherent bond formation takes place between enzyme and product complex. And now in the third step, newly formed product is released by separation of enzyme, and same enzyme is used or recycled for first step. So this is very important steps that which enzyme provides the reaction between substrate and enzyme, and rate of product formation from the substrate takes place. This is the mechanism which is occur in various kinds of enzyme catalyzed reaction. Now see, what are the enzymes? Enzymes are having catalytic effect. The equilibrium concentration of product and reactants, they only increase the rate of reactions, and therefore equilibrium is reached more quickly. Enzymes provide the effect of equilibrium concentration of product and reactant likewise, and as a catalyst they include enzymes catalyze reaction, and how the reaction takes place we will see with the help of diagram in the next one. Energy activation diagram for catalysis. So enzyme catalysis reaction takes place. Okay. Now this is about the mechanistic part. Now we will see how this reaction takes place. This is the activation, energy of activation or progress of the reaction shown by diagram. On the graphical representation, there are two axes. Vertical axis, that is free energy, keeps free energy. Horizontal axis shows progress of reaction. Now free energy of reactant, that is two reactants shown in the graph. Whenever reaction is not catalyzed by any enzyme, it is shown by a regular graph, that is having higher activation energy, which is without catalyzed, requires more energy, and shown by this arrow at topmost position. So whenever two substrates reacts with each other without any enzyme catalyst, then required transition state requires more activation energy. No doubt product is obtained, but rate of reaction is slow, as well as it requires more energy of activation so as to form its transition state. But whenever the same substrate react in presence of enzyme catalyst, in the graph curve is shown by dotted lines. It requires low activation energy and transition state is formed easily. No doubt we are getting same product, but enzyme catalyzed substrate reactions require lower active energy of activation and rate of reaction becomes much more higher. But in case of without enzyme catalyzed reaction, energy of activation is higher to form a transition state, and that's why rate of reaction is get slower one. So this is one of the graphical representation by which enzyme substrate reaction takes place. Okay, thank you.